Hi, my name is Frances Wong. I currently lead the IDR project, and I'm happy to be giving you this year's update on IDR. For those of you new to IDR can be found at idr.openmicroscopy.org. You can see that we have cell IDR and tissue IDR. You can browse all studies in IDR or choose to look at cell-specific or tissue-specific studies. You can also query IDR by a variety of options, such as IDR number, gene, antibody, compound, or organism. To show you an example, we will do a compound query towards the end of this presentation. When you have time, please have a look at what IDR has to offer. IDR believes in making data fair, that is, findable, accessible, interoperable, and reusable. Findable, where the original image data is cross-linked from a publication by DOI. Accessible, where the metadata is retrievable and can be queried using an open API. Interoperable, where we use common defined vocabularies such as public ontologies. Reusable, where we strongly encourage the sharing and adapting of data under an open license. But which datasets should IDR make fair? Unfortunately, we can't do this with all datasets as it's a time-intensive process. We have therefore chosen to focus on reference datasets submitted by the community. This means we look for valuable images from each domain that either have links to existing resources or where we can help to establish those links. At IDR, we also work with other resources to ensure that datasets can be published in the most effective, appropriate ways, with the goal of maximising utility and reuse. In this case, IDR works with the Bioimage Archive to provide public archiving of bioimaging data, thus building what we call the bioimage ecosystem. So bioimaging datasets with minimal metadata are deposited at the Bioimage Archive, and bioimaging datasets with reference metadata are submitted to the IDR, where we provide substantial curation and thus added value to these datasets. In addition, reference datasets in the Bioimage Archive come to IDR, and IDR datasets are archived at the Bioimage Archive. IDR currently has 261 terabytes of imaging data, consisting of 29 million image files, over 89,000 genes, and 40,000 compounds. In reality, this translates to over 85 studies, cross-published in different journals, and cross-referenced via accessions and DOIs. IDR contains integrated studies with thumbnails, experimental metadata, biomolecular annotations, and analysis results. The whole IDR system is run and stored at Embl EBI, allowing users to browse data, perform cloud analysis, and download for local analysis. Looking more specifically at the metadata. Metadata in IDR is curated. Experimental metadata is data generated by the image acquisition system. Biomolecular annotations provide added value to imaging data as it describes structural, functional, and phenotypic features of genes and proteins, which effectively helps the biological interpretation of biomolecular results. Analysis results reports the analysis performed by the submitter and reveals the results found. Looking at linked metadata. In IDR, we group attributes together, such as genes and phenotypes. We see here in the example IDR 12 that there is the phenotype of elongated cells associated with the ASHTL gene. If you click on the ASHTL gene, it will show you a list of studies with results for this gene, and thus linking between studies. The elongated cell phenotype has also been annotated with a CMPO ontology term, where CMPO is a cellular microscopy phenotype ontology. Ontology terms allow submitters to normalize phenotypes. So different submitters may express very similar phenotypes in different words, but we can use the ontology to link them to a common term. So searching for the elongated cell phenotype, CMPO term, we can get back studies with submitter phenotypes relating to the elongation of cells. This leads me on to ontologies. A common question that people often ask us is, what ontologies should I use? We don't have a definitive answer for this, but I can tell you the public ontologies we're using in IDR. For organism, we use the NCBI taxonomy. For study type, high content screen types, and protocol, we use the EFO. For imaging method, we use the FPBI, and for phenotype, we use the CMPO as previously mentioned. For gene, we use either ensemble or NCBI gene, and for protein, we use UniProt. Recently, IDR has been receiving more clinical data on compound screens. So we have been using SNOMED CT for clinical data and PubChem for compounds. Those of you who used PubChem will know that it's a chemical database rather than an ontology, but we wanted compounds in IDR to link to an authoritative resource that is non-commercial. There are, of course, other chemical databases such as Chembl. So some compounds in IDR have IDs for both PubChem and Chembl. Other important compound fields to highlight are compound smells and INCHI keys. However, all this is work in progress in IDR, and we're working with the Chembl team at EBI to try to standardize the metadata for compounds. Now let's have a look at some image data, starting with one of the largest datasets in IDR. This is a tissue dataset from the Human Protein Atlas, published by Yulin et al. Under IDR 43, you can see high-resolution human tissue sections where protein expression has been mapped across all major tissues and organs in the human body. We currently hold 9,000 antibodies from the Human Protein Atlas, which is about a third of the total antibodies we expect to receive. So this is a growing dataset in IDR, where we will release additional antibodies in batches. Moving on to some cell data. IDR 47 is a yeast single-cell transcription profiling dataset from Gregor Newert's lab. The goal of this study was to generate datasets for single-cell transcription modeling. If you select one of the images, it will open up in the iViewer, and you can see that it has many regions of interest. When you click on Show, you'll see that this image has 372 individual masks, and you can deselect or reselect the mask you wish. Just to quickly mention, this dataset is an example of a study that has been archived in the Bioimage Archive. You can hear more about the Bioimage Archive from Matthew Hartley's presentation. Moving on to IDR 83 from Lamars et al., which is the first SARS-CoV-2 dataset that IDR received. Here we have detailed electron microscopy images showing various particles in a human organoid, where the image viewer allows you to zoom in and view the image up close. The trained eye will be able to detect the onset of virus infection at different stages of the viral life cycle. We have co-published these images with our colleagues at Empire to provide public archiving of this dataset. And you can hear more about Empire from Arden Pardwarden's presentation. Moving on to another SARS-CoV-2 dataset, IDR 94. This is a large-scale drug screen on human cells to identify inhibitors of SARS-CoV-2 by Ellinger et al. At the Fraunhofer Institute. Taking well A1 as an example, we see the cells were treated with the compound loratadine. For the hay fever sufferers amongst us, we know that this is the antihistamine sold under the brand name Claritin. Here in the compound supplementary, we see the PubChem ID, SMILES, and INCHI keys as mentioned earlier. I also mentioned that we would do a compound query, so let's search for the antiviral drug remdesivir. The results show that IDR has 260 images with the compound remdesivir in one study, IDR 94. Now looking specifically at these images, we see a nice dose-response curve as we start with the highest concentration of remdesivir at the top and finishing with the lowest concentration at the bottom. The activity of this compound on this human cell line is clearly visible. There are many compounds in this screen. Please feel free to have a look. Its accession number is IDR 94. Moving on to Open Cloud and Open Analytics. One of IDR's goals is to integrate well-annotated studies and allow data mining and data reanalysis in order to discover new biological findings and gain further knowledge. This can be done using interactive notebooks, such as Binder or Jupyter notebooks. This example shows a new IDR notebook exploring the IC50 of compounds from IDR 94 screen B, the SARS-CoV-2 data we just looked at. Taking remdesivir again, we have calculated an IC50 value of 0.81 micromolar, which is close to the reported value in the publication of 0.76 micromolar. The next step is to plot the dose response curve. And here you see the dose response curve for remdesivir. This notebook can be used to calculate the IC50 value and plot the dose response curve of other compounds in the screen. You'll have an opportunity to explore this notebook in more detail during the IDR workshop. A second type of reanalysis is the imaging data itself. Currently, in IDR, the raw data for each published study can be downloaded using Aspera, and then users can perform their own reanalysis locally. However, this approach comes with its own set of limitations. So in the last year, the OME team have been working on ways to allow scalable access to the binary data in IDR. The image on the right shows a preliminary result of this work. Taken from the study IDR 62, this is a 5-dimensional image which has been exported from IDR into the ZAR format and uploaded to an object store at EBI. The image is then remotely accessed from a client in Dundee. In this case, it is loaded and visualized using the multi-dimensional image viewer Nipari. This is still work in progress, but you can find out more about it from the recent preprint by Josh Muir et al. Now, to quickly summarize, the IDR is publicly available, curated studies submitted by the community in a searchable, scalable platform that links metadata and enables reanalysis that can be deployed by others. This just leaves me to thank our funders and everyone on the IDR team. Thank you.